degree in oceanography in Oregon State University. And he decided to stay for his PhD in the West Coast, but elsewhere in the University of Washington. That was a PhD in fisheries. Uh, and then he went on to do a postdoc position at a Pacific Biological Laboratory, guess where? In the West Coast as well, but like more northern in British Columbia in Canada. And after his postdoc, he started working for NOAA, and he focused on the early life history and recruitment dynamics of the walleye pollock, which is an important fishery in this part of the world, and especially in the Gulf of Alaska and the Bering Sea. And he started working for the Alaska Fisheries Science Center over there, that very cool area. And I guess then he came back to Oregon, because Oregon is beautiful. Yes, to the research. Yeah, that's it. Of course. And he started working on the research fisheries oceanographer as a research fisheries oceanographer in the fish ecology division down in Hatfield. I don't know if you guys have had the chance to visit, but there's a lot of cool oceanography happening at Hatfield in the joint campus that NOAA and Oregon State have over there. And his work has been like particularly relevant for many of us in the Sunland Lab, because he has worked a lot on feeding and food web interactions between zooplankton and fish. And Ric has this great understanding that jellyfish are also an important part of the ecosystem, because he really likes jellyfish and has worked a lot in also gelatinous zooplankton trophic ecology, and how they interact with fishes, right? So we read a lot of his work. And he has also co-organized 5 sessions at major scientific meetings. And it's interestingly enough, because of jellyfish, right? A lot of fisheries biologists don't pay that much attention to jellyfish, but Ric has had that like unique vision in also work like focusing on those organisms. And yeah, like I personally want to say that when I first came to the States, it was great to meet collaborators of Kelly like Ric, which are very approachable, and we're really happy to share information and to help other students navigate the complexities of academia. So yeah, I'm very happy to have you here, Ric, and I guess you can take it away.
Okay, thanks, Marco. And I should say it's been a pleasure working with you and others in Kelly's lab, including Kelly. So it's great. And hello, everybody. So I'm going to start off and talk a little bit about some of the recent work we and my collaborators have been doing here on pyrosome blooms and their effects on the ecosystem. So, but I want to just turn off my video here just so, because we're having problems with bandwidth. So there you go. Okay. So anyway, hopefully you can still see my screen here. But so first of all, part of the talk is on marine heat waves. And I'm sure that a lot of you are familiar with marine heat waves, similar to heat waves on the on land, but they tend to be much more prolonged and often even greater intensity relative to the norm than terrestrial heat waves. They've been described by their duration, intensity, the rate of evolution, and particularly by their spatial extent. And more recently, they've come up with a sort of a classification system that similar to what we have for earthquakes and hurricanes, where they have multiple levels of from sort of moderate all the way up to extreme. And this is sort of just a little diagram showing if this was sort of your normal climatology, the line here on the bottom, and you had a year that looked more like this in terms of the temperature and how many standard deviations it is from the normal, you could see that sometimes it can get to be extremely warm, up to 4 standard deviations above the normal temperature, which would be classified as an extreme heat wave. So there's been a number of heat waves that have occurred throughout the world's oceans. See, Australia's had a lot. There's been some in the North Atlantic and Mediterranean, but by far the biggest one has been the one that has occurred in the North Pacific, here in 2015 and 2016. You can see somewhat the spatial extent of that. And how this all started was, as you know, many of you know, the North Pacific can be a very stormy place in the wintertime. And generally there's a lot of mixing of deep water, cold deep water up to the surface But as, there was a very unusual atmospheric condition that happened in the winter of 2013 to going into 2014, where there was this high pressure ridge that prevented all that mixing from occurring, so that it resulted in all the warm water that had produced the previous summer just stayed up on the surface. And so this is an anomaly plot that shows the difference in temperature from the normal situation. You can see there's a really warm blob of water up there. And these authors actually came up with a very scientific name, calling it the blob. So that's kind of the name that is stuck in the literature. And you've probably heard about the warm blob quite a bit in reading any kind of newspapers and things like that. So, and this is just this sort of quarterly temperature maps anomalies of the North Pacific going from July of 2014 to August of 2017. And of course, the red is warmer than average. And you can see during this time period, there's a lot of red in the North Pacific. It was very, very warm place to be. And of course, the blob was sort of the, originated even before this time, but it was very big around the middle of 2014. And it continued and pushed actually close to shore here. But actually come around October of 2015, there was a very strong tropical El Nino off the coast of South America. And that continued to push up warm water up the coast here. So we had sort of two things going on, the blob and El Nino, that made unusually warm conditions in our region. And just to show you, for much of 2014, the blob is actually out in the middle of the Pacific, so it really didn't affect the California current. But this is from a buoy that's about 36 kilometers off the coast, so almost at the shelf break here. And this is showing sort of a seasonal cycle, the normal anomalies in temperature. So they, maybe fluctuates a couple degrees above and below the normal temperature. But all of a sudden, in September 14th at 10 p.m. to be exact, this incredible warm blob of temperature came on, warm water came onshore. So the temperature actually went up almost 6 degrees in a matter of just a few hours, which is almost unprecedented in our area here. You never see that. In fact, the 6 degrees is probably almost what we get on an annual a range of temperature. So this was a really anomalous situation, because that's when the warm blob came onto the shelf, and it slowly got a little bit cooler, but it really was been above average temperatures for most of that time period. And this is for based on satellite data from within this little box off the coast of Washington and Oregon, and this is showing. So the blue line is sort of the climatology, the typical seasonal pattern. The black is the actual data from that time period. And you can see for a period, it was from here all the way up to basically here. It was in almost marine heat wave area. It was definitely one standard deviation, which is this green line above the average, and sometimes 2 or 3. So it was really an unusually warm period. And this actually has, this heat wave has been classified as a severe marine heat wave, so the second from the highest level, but it had a duration of 711 days in the central North Pacific, which was more than twice as long as any previous recorded marine heat wave. So as you can imagine, with all this warm water out there, there were probably a lot of ecosystem anomalies, and there certainly were. I'll just give some examples of some of those. There was a dramatic decrease in the overall primary production off the coast of, off the west coast, but there was actually an increase in harmful algal blooms, which led to lots of cases of shellfish poisoning and closing of shell fisheries along the coast, including the crabs fisheries. So very big impacts on fisheries. There was occurrence of many tropical and offshore copepods and other higher trophic levels, and a lot of decreases in the normal taxa that we see off of here. There's a lot, we noticed a lot of changes in reproduction and growth of fisheries. For instance, in reproduction, the phenology change, where we had species that usually spawn in the summertime off our coast, like anchovies and sardines, were actually spawning in the winter because the temperatures were more typical of their their summer spawning conditions. So there was a real change, and that had a lot of implications for the ecosystem. There was a lot of unusual mortality events in both marine mammals and seabirds. and dramatic changes in their distribution. A lot of the marine mammals were pushed very close to the shore where there were a lot of whale entanglements and things like crab gear that we usually don't see. But for me, the most interesting thing is that there was major changes in the gelatinous zooplankton, where we had decreases in a lot of our normal Scyphomedusae, the big, large medusae that we normally see off here, but an unprecedented bloom of pyrosomes in the North Pacific. I think this, to me, is the real poster child of this warm blob here, and I'll talk a little bit more about that. So a lot of you probably have heard of pyrosomes, but if you were like me a few years ago, you probably didn't even could barely say anything about or knew anything about their biology or anything like that. So this is an example of what probably the general public has seen of pyrosomes in the last few years. They've had lots of occurrences of big, massive die-offs, and they wash up on the beaches and litter the beaches for miles and miles. But sort of a more scientific thing, there, as you're probably familiar, there are a number of different gelatinous zooplankton groups. Most of you are probably more familiar with the medusae, the large Scyphomedusae and hydromedusae, a lot of which Kelly and her group work on. There's also the comb jellies or ctenophores, which is another phyla of jellies. But there's one group that's kind of different, and that's the what we're called the tunicates. So these are pelagic tunicates related to the sea squirts, and they come in a lot of different shapes and forms, and probably most people are not as familiar with these because they tend to occur further off the shore in more blue water. So the pyrosomes are one of those groups. There's about 23 species worldwide. But one of the things that's kind of interesting is that the typical jellyfish you think of, the medusae and the comb jellies, are actually very primitive evolutionarily. They came off as early on in the evolutionary tree of organisms, whereas the tunicates, as you probably all know, are closely related to the vertebrates. In fact, one of the last groups to break off from the vertebrate line, and in fact, the most closely living invertebrate species to humans are in this group. So they are very closely related, so quite different evolutionarily from the typical medusae. And then of the 23 species, there's one species that's probably the most dominant in the world. It's called Pyrosoma atlanticum. And it occurs in pretty much cosmopolitan throughout tropical and subtropical ocean waters. And as this is a map developed a few years back that shows the distribution of pyrosomes from sampling around the world. And you can see that most of their distribution occurs between 50 degrees north and 50 degrees south. So the warm belt of the tropics and subtropical regions. You can see they do get pretty far north up in the Atlantic, which tends to be warmer than the Pacific at the same latitude because of the Gulf Stream. But in the Pacific, they've only been found to sort of like central, maybe Southern California here. Certainly not up in our area, which is kind of highlighted here as a star. So this is something that we haven't seen before and we wouldn't expect to see up in our area. But it happened, and what I'll be talking about today is mostly what I'm calling the Great Pyrosome Bloom in the North Pacific Ocean. And I'll talk a little bit about how it came about, its effects and implications for ecosystem. So probably a lot of you may have seen a lot of news reports. It was all over the place for a while that this was a big thing. In fact, we were getting reporters calling us on a daily basis from all over. So it was just amazing that the amount of publicity this got. And they often, I guess the common name that they've been using was sea pickles because they kind of look like large pickles here, translucent pickles. But anyway, there was a lot of news coverage of this. So what I'd like to do is talk about how, at least in our sampling, we first observed these pyrosomes. And to do that, I'll describe sort of one of the cruises that I was lead of before I retired last year. And this is what's called a pre-recruit survey, where we go out and sample for juvenile fishes to kind of get an idea of what the incoming year classes might be, which might help management. But at the same time, we caught a lot of other different invertebrates and squid and things like that. So we tended to enumerate and all the taxa that we found. So we had a number of cruises, mainly during the May-June period. We sampled with a very large net, but had very fine mesh in the back of it. And we did a whole bunch of other things along with it. And we basically sampled all these lines along the Oregon coast and one up into the Washington state here. And just for perspective, here's the Newport line, which is regularly sampled off of Newport. So anyway, so what I'm going to do now is show sort of the catch of pyrosomes in these trawls. And these are log abundance, and there's a scale here for blue being 0 all the way up to high. And then there's a number up here that is the mean catch of pyrosomes in the hull on a 15-minute tow. So you look at the first couple of years shown here, 2011 and 2013, you can see that there were basically no pyrosomes. And I think if you had done this survey 50 to 100 years previous to that, you probably get the same result. However, we started seeing what we thought were the first pyrosomes in 2014 at the southernmost part of our survey. 2015, 2016, they increased by orders of magnitude. And then by 2017 and 2018, they were by far the dominant species we collected on our trawls. And they were so I mean, you can see here, we got almost 12,000 in a 15-minute tow, way more than anything else we got. And it was getting so bad that we could not even, we had to stop our survey. We could not do these northern areas because there was just so many pyrosomes in water. The nets were ripping up and we just couldn't do it. So anyway, just a really big change in the ecosystem. And just to kind of put this in a little larger perspective, so what I showed you are the sampling from these two upper boxes here, but there's a group from our Santa Cruz lab in California that's done very similar sampling all the way down to Southern California. And this is sort of showing a time series. And in fact, this area, oops, I'm sorry. So this area C here, shown here in black, they actually sampled since 1983. So this is kind of off of San Francisco Bay down to Monterey Bay. And you can see that occasionally they got a few pyrosomes in the sampling during that time period, but not typically. But you can see what happened, but not only that, but all the areas had huge numbers of pyrosomes going up to the present. And in fact, Oregon had some of the highest, the ones in blue had some of the highest numbers. So again, this is a log scale. So these are really big increases in pyrosomes. And here's just to show you spatially how this looked. So this is for 2 years here, 2013 and 2014. Again, this area up here is the area I showed you earlier. So you can see they started showing up in our area in 2014, but they were already starting to move up north and off of California. And then by 2015 and 2016, they were pretty much all the way up and down the coast. The star indicates where the highest catch of the whole survey was. And you can see that that was actually up off of Oregon. But looking at even subsequent years, 17 and 18, they were just everywhere off of Oregon. Literally every catch had some, and they were in huge numbers. They actually started decreasing a little bit off of California those years. But then 2019, they started moving back down south, and we got only one pyrosome, I think, in our sampling in 2019. So really interesting change in their distribution, probably related to the heat wave. And then kind of looking even a bigger perspective, we analyzed trawls from a lot of different surveys from a couple off the West Coast, one off of Canada, if that's Vancouver Island, and then even all the way up into the Gulf of Alaska. And it turns out that there were pyrosomes caught way up here in the northern Gulf of Alaska. So an incredible range extension of about 3,000 kilometers from their previous known range. So really incredible change in their distribution. So now I'd like to sort of give you something, a little bit of talk about the biology of pyrosomes, and just to kind of give you a look at their anatomy. They're actually colonial organisms, so you can see that they're made up of a lot of different individual, what were called zooids. These are all genetically the same within one individual, and you can see that they're all orientated so that they have an inflow and a hole on the outer side that takes water in. They filter through their atrial basket here, and then it comes out the other side into the central part. And they use this to not only feed, but also to propel themselves. So the water gets shot out and they can move in a various direction. And you can see these are examples of zooids. There's a little bit of difference in the morphology, but they're fairly similar. And this is showing sort of another thing about their feeding. So you can see their the phytoplankton are taken in and they're retained inside their filtering apparatus, and pretty much clear water is sent through and they are propelled beyond that. So they are known to have very high clearance rates, and they also are known to feed on very small microorganisms, less than 10 microns, or in another study, 3 to 5 microns, and even are feed on bacteria at the submicron scale. So we had, we were lucky enough to get some funding, and we got some emergency sort of rapid funding from NSF to look at this. So I was involved with a project from people from Oregon State University. And also I was worked with Kelly Sutherland and others from University of Oregon. We got a Sea Grant project funded. So that was great to be able to work collaboratively with different groups on this. And so we had a couple of different goals to look at their distribution in relation to environmental variables during a couple of years. And also we were trying to we wanted to characterize their diet using sort of chemical biomarkers, fatty acids and stable isotopes to look at, and then also estimate their feeding rates and trophic impacts. So this is showing the sampling we did for 3 times, February, May, and August. I'm sorry, 6 times actually in 2 years, 2017 and 2018. Those are 2 very big years of the bloom, if you recall, up in our area. So we sampled a number of different stations and then went very far offshore on one and used a lot of different plankton gear to collect them. And this is showing sort of the size distribution for February and March, shown over here, and in May. So you can see that they're basically doubled in length over that time period. But the interesting thing was that we didn't see any real changes in their density. So either the mortality was balanced out by new recruits, or there wasn't a lot of export for the population over that time period. So again, this is showing the distribution during 2 cruises, May and then August, up here off the coast of Washington and up into British Columbia. And you can see that they were found at most of the stations that we sampled. And a lot of this has been written up in a paper that was led by Julie Schramm, who was a postdoc with Aaron Galloway and Kelly over at OMB. Anyway, we looked at the distribution of their in relation to temperature, and you can see that they tend to prefer the higher temperatures, the warmer temperatures. You see a little bit of a pattern with salinity, but it's sort of more intermediate salinities. And then the interesting thing was for in relation to fluorescence, they didn't. There wasn't a significant relationship, but there was definitely a very strong threshold above which there were no pyrosomes found, and it's probably because at higher chlorophyll levels, they're probably too much productivity, and they don't do very well in sort of highly productive coastal waters. So basically, they're found at the kind of the same area that in the tropics, but just happens that that kind of water was found off the coast of Oregon and Washington in the last during the marine heat wave. There were a number of studies that looked at their vertical distribution, and this is one that was done by a REU student of Kelly's and mine, Olivia Blondheim. She worked with the Oregon Department of Fish and Wildlife, has this bass camera system that they put down and look at fish underwater, and it was great for measuring. It's a stereo camera, so they can measure both the numbers of pyrosomes, but also their orientation, their size, and everything. So I looked at a bunch of different stations off the coast here. And just to kind of show a brief result, we see that they were basically found in midwater between about 25 and 35 meters. About 71% of the pyrosomes were found in that area during the daytime, at least. We think they come up close to the surface at night, but we weren't able to sample with this gear at night. Another example, that Hillary Sorensen, a lot of you may know, is a U of O graduate student, recent graduate. So she put a GoPro cameras on a CTD frame and picture of them in the water column. And she saw that the highest densities were a little deeper, between 49 and 68 meters during the daytime, but below the thermocline, above the pycnocline, and actually definitely below the higher chlorophyll areas of the water column. So next well, actually Hillary was involved in this, where she was looking at using fatty acid biomarkers to look at their diet composition. And one thing about, because they're eating such fine particles, it's very difficult to look under a regular microscope and see what they're eating. So we ended up by using sort of biochemical means. And probably a lot of you are familiar with essential fatty acids. Those are ones that come from their food source. So by taking a measurement of the fatty acid composition or signature of a organism, you can sort of tell where their food came from. And a lot of the basal producers, such as green algae, brown algae, different phytoplankton, all have a different fatty acid signature that you can see that gets transferred to their predators or grazers. And this is just an example of some of the results we found, that there were some seasonal differences. So this is May versus August of the different, and these are the different prey groups. You can see bacteria were pretty well represented, and dinoflagellates as well. They're sort of the two dominant groups that were represented. Diatoms, which are the normal dominant phytoplankton off here, were barely represented at all. So they're not able to probably feed on such large planktonic organisms. Also, I wanted to highlight, and this is something I wasn't involved with, but Kelly Sutherland and Anna Ward were just had a new paper that just came out. Here's it was led by Ann Thompson from Portland State University, shown here. And they looked at a bunch of different techniques, including flow cytometry, environmental SEM, and also they did genetic sequencing and found some really interesting results. This just came out last week, and encourage you to take a look at the paper, but showing that they're eating fairly fine, small particles, but something, including several different kinds of bacteria were found in there. And the interesting thing was, based on the genetics work, they were able to show that the pyrosomes harbor a different microbiome community than the surrounding seawater. And there were some actual new taxa that they discovered. So it was kind of an interesting that they are actually providing perhaps producing different microbiomes that are brought into our water from being transported there. So it's kind of an interesting study. So the other thing we looked at was stable isotope analysis, and I think a lot of you are probably familiar with this method, but it uses usually you look at carbon and nitrogen as the two main isotopes, and carbon gives you an indication of where the basal food production is. So you can get sort of an offshore signature in the carbon, and then you can also get an inshore, you'll get a different one in an estuary and stuff like that. So it can sort of give you an idea of where their food production is coming from. And the nitrogen is interesting because it increases by about 3.4 parts per thousand for every trophic level you go up. So by measuring an animal's nitrogen ratio, you can tell basically what trophic level it's feeding at. So we had some results from that in this paper as well, where we looked at different locations and different, both in north and south, and then inshore, offshore by depth. And we see some variation in the signatures there for both those parameters. But I think the real interesting thing is, if you look at, for instance, nitrogen, this red line shows where a typical copepod might be at in our waters. And then the green is sort of a typical diatom or other phytoplankton. And that again is about 3.4 parts per thousand difference between those two. But the interesting thing is that most of the pyrosomes are actually feeding at a lower trophic level than the copepod. So a real, they're really at the base of the food web here. And then the last thing we kind of looked at in this study, well, it was with the NSF-funded study, was we wanted to try to look at their ecological impacts. So by measuring their ingestion and grazing rates. So to do that, we looked at gut fluorescence over a diel cycle, and you can see here the pattern, and they tend to have a higher average gut pigment at night. So that's probably when a lot of the feeding is going on. We managed to keep them alive and on tanks on deck, and we're able to measure their gut evacuation rate, so how long the pigment is found in their guts. So by putting those two together, we could estimate a grazing rate, how much consumption they do of chlorophyll. And this is showing by different transects. And this is the Newport line here, which had the highest grazing rate of all the different transects we looked at. And then finally, by measuring the sort of ambient chlorophyll in the water column, we can estimate their grazing impact, what percentage of that is consumed. And again, by different transects, you can see the highest was about 12% per day was consumed off of Newport. So pretty high impact by these guys on the water column. And just to kind of show you a more geographic perspective on that, you can see there was a lot of variation. So this is showing for the 3 different cruises, February, March, May, and September, the catch of the pyrosomes in the upper panels, the grazing rate in the middle, and then the grazing impact. So just looking at the grazing impact, you can see it was very low in September. There were hardly any caught at that time. Somewhat low during the February, March period. And then May had, some mostly low, but there was one location that was extremely high off the coast of Oregon, off the coast of Newport, I should say. So there seems to be some areas where their impact may be very, very high. So the other thing we were interested in is where did all these pyrosomes end up? They're, incredible biomass. There was estimates of 200 kilograms per kilometer cubed during some of our sampling, in densities of 5 individuals per meter cube. So if you think of a meter cube box, there'd be like 5 individuals, which is pretty high density for large organisms like this. The caloric content was measured to be around maybe 5 kilojoules per gram dry mass, which it's higher than a lot of other invertebrates, especially a lot of the crustacean, higher than almost all the other gelatinous zooplankton. But it was still well below the range of other forage species, mostly forage fishes that you can see here, probably 2 to 3 times higher. So it's not the best prey, but certainly not the worst either. And we don't think there's not a lot of evidence that these pyrosomes only live to be about a year, and then we think, and then they die, and we don't think that they're actually dissolving in the water column. So either the biomass was consumed by predators, or it dropped down to the bottom to enter sort of the, to be remineralized and enter the detrital pool. So we actually did a little survey of
Rick, it's cutting down.
We lost your audio, Rick.
Yeah.
Not sure if you...
I'm sorry, Kelly?
We lost your audio for a minute there, but now I can hear you again.
Okay, I'm sorry.
Right about when you switched to this slide.
Okay, I'm sorry. I'll repeat it. Thanks. Okay, so we were interested in looking at potential predators of it. So we did some survey, and we found that there were indeed a lot of species of fish, including juvenile and adult salmon, halibut, tuna, and a lot of different rockfish species that were consuming them. And then there was actually even an example of a fin whale that was caught or that actually washed up on the beach off of Washington, that had his stomach actually full of pyrosomes. There was nothing else in the stomach. And the whale was very emaciated. We don't know if that was the cause of their death by eating too many pyrosomes and not getting enough other things, but certainly wasn't doing very well. And this is an example for a lot of the fish predators. You can see these are locations where we had diet data off the coast of Washington, Oregon, and Northern California here. So each one of these represents a location where we had diet information. And there was only, from the period 2004 to 2014, there was only one location, which you barely see here, where we actually found the pyrosome in the gut. And that was during 2014, which, as you remember, is when they first started appearing off our coast. But then if you look at the sort of the data from 2015, you see a lot more occurrences, 2016, and by 2017, pretty much not every station, but most of the stations had predators that were feeding on pyrosomes. And in many cases, it was the vast majority of their diets. So there was a lot of consumption of pyrosomes by many different fish species. There's also a lot of invertebrate species that were feeding on it. These are just some examples from a cruise I was involved with, where we had an ROV that went down in the bottom of Quinault Canyon, so about 1,000 meters down. And these are just examples of, this is a sort of a bathypelagic jellyfish that was feeding on a pyrosome. You can see it hanging from its tentacles here. There were sea urchins that were feeding on them, sea anemones, a whole bunch of crabs, different things like that. So apparently a lot of things in the benthic food web were able to take advantage of this food source. There was also, but a lot of it just ended up by sinking down to the bottom. And there's examples from a paper that Kelly led that showed high numbers of jellyfish from ODF and WROV surveys off the coast here. Another one from that were found in the bottom of Quinault Canyon. But also a lot of them tended to wash up on the beaches, as I showed you. And this had a big potential for big input of carbon to the sort of terrestrial ecosystems. And many birds would feed on these, insects and other things that were living in the sands of the beach would be feeding on them. So it had a potential to move, out of the ocean and actually onto the shore. And one of the interesting things was that there was some occurrences of fish that were found inside these the tubes of the pyrosomes. As you know, they're sort of a hollow tube with one opening. And we actually had documentation of two species, rockfish, a little juvenile rockfish up here, and then another species called the medusa fish that was living inside, or at least were found inside these pyrosomes. And we don't know, obviously that's probably a benefit to the fish, be able to avoid predation in the pelagic zone. I'm not sure if it helped the pyrosomes at all. So it's probably more just commensalism and not any mutualism involved, but certainly an interesting thing. A lot of fish do live with gelatinous zooplankton, but seldom do you see them living inside them like this. There were a lot of negative interactions, of course, and particularly the fisheries were very hard hit. This is an example of shrimp, salmon trawling gear that was just absolutely covered with pyrosomes, and took a lot of work to clear these all off when they came aboard. This is an example of a shrimp trawl that, so you can see these little red things are the shrimp, and it was just full of pyrosomes. All these little brown things here are pyrosomes and some number of fish. So it meant that the fishermen had to clear out all the pyrosomes to find all the shrimp. So probably led to a lot of extra labor and maybe some lost catches. But the other thing that it impacted as well is, as I mentioned, it impacted surveys a lot. Our surveys were heavily impacted. We would catch huge numbers of pyrosomes. And here's an example of a catch here where we had to kind of go through and pull out the few little fish that we were looking for. And it just made it a very difficult situation. And as I mentioned, we actually had to cancel some of our hauls. So it was very unfortunate that we just couldn't do anything about it. There were just so many pyrosomes out there. And I wanted to put this up. This is a paper by Keats Conley, and then a graduate student, former graduate student from U of O, and Kelly. It just shows kind of the importance of these mucus mess graziers. So you can see pyrosomes here along with the other pelagic tunicates are very important in the pelagic food web, in that they're able to feed on relatively fine particles and transfer some of those up to higher trophic levels, although not all of them do get transferred up. But they also are very important in sort of the ocean carbon cycles, that they produce fecal pellets that drop to the bottom of the ocean. And also they have a lot of jelly falls. Basically, the dead carcasses fall down to the bottom and sort of become part of the benthic food web. So these are very important parts of the food web here in many, particularly more oceanic ecosystems. But because of the marine heat wave, they ended up by being very important in our ecosystem off the coast here. So in conclusion, pyrosomes were found in warmer, less productive waters in the NCC, which is somewhat similar to what they're found in their normal habitat, the tropical Pacific pelagic habitat. So it just happens that they probably came in with the marine heat wave. Conditions were very good, favorable for them, so they they grew very quickly and pretty much overwhelmed the ecosystem, I think, in terms of their abundance. They feed on very small planktonic organisms, and they have very efficient feeding and clearance rates. So they could be potentially depleting food for other plankton. Now, we don't have a lot of evidence for direct evidence. As you know, this is sort of a typical food web off our coast from phytoplankton to zooplankton to maybe jellyfish, but hopefully to a lot of forage fish that may be eaten by higher trophic level predators here. However, when you have pyrosomes, it sort of creates a new sort of trophic web that doesn't go through the traditional part. So we don't know how this affected the ecosystem. But one thing that we don't have a lot of information on what other pelagic crustaceans are eating. That's the normal dominant planktonic organisms that we have off our coast here. But we do know, we do have some data on their changes in their abundance. So these from that same survey I showed you earlier, these are the catches of krill or euphausids in all our different hauls during the first 3 years shown here. And these numbers are actually a whole different scale. These are in hundreds of thousands or tens of thousands. So this is 41,000, 178,000, 175,000. So this is normally the dominant species we get in our surveys or dominant taxa. However, once the marine heat wave came in 2015, 16, and 17, and 18, you see a real drop off, orders of magnitude decrease in the abundance of krill in these waters. And you can see that there were stations with almost no krill at all, which was totally unheard of prior to that time. So this has very important implications for the ecosystem because krill are, as you know, one of the dominant food sources for not only fish, but many seabirds and marine mammals feed a lot on krill. So not having a lot of krill out there is really a big deal. And not only were there not a lot, the ones that were there were generally much smaller than normal. So the biomass decreased dramatically, not only the abundance, but the biomass was much lower. So they could be potentially a food source for other higher trophic levels, although we don't think they're quite as good as some of the other available food sources out there. So it's something that it would be worth looking into more in the future, but particularly if they're continue to be in our waters. And that brings up a question. Are pyrosomes going to be permanent residents in the California current? Well, we don't know that, but there's some evidence that show, well, I should say modeling studies that show that based on, depending on which climate scenario you're looking at, you can get anywhere from sort of like 40% to maybe 80 to 90% of the world's ocean will be in a permanent state of marine heat waves. So that means that the full 365 days are going to be above the minimum threshold that was established for heat waves. So basically it's going to be, setting a whole new threshold basically, but there are some areas, but like the North Pacific that may reach that by 2040, as you can see here in green. So that's not that far in the future that we may be in a permanent marine heat wave. So, and just as an example, this is a temperature anomaly plot for that blob year, 2014. You can see all the warm water in the North Pacific. Again, there's just a little bit, there is some cold water close to the coast in the upwelling area, but pretty much the whole North Pacific is covered with warm water. There was actually another marine heat wave that came in 2019. So this is showing basically September of 2019, not quite as strong as this one, but still fairly intense throughout much of the North Pacific. And again, in 2020, there was also even a bigger one than 2019 throughout large areas in the North Pacific. So we may already be heading to a point that at least we may be getting marine heat waves for a good part of the summer. And this is another plot from this blob tracker website that you could go to get more information. But this shows sort of the area of extent of sort of the warm events in the North Pacific going back to when satellite imagery first started back in the early 80s. And you can see that there have been a number of events such as the 82, 83 El Nino, which at the time was huge, but obviously it didn't cover a huge amount of area. There was a 97, 98, which was even bigger, but again, it did cover a lot, but it's not as much as others. There was a warm period in 2004, 2005 that also had pretty good coverage, but the big thing was if you look at the blob and how not only how long that lasted, but how much area it covered. It's huge. But again, there was also sort of little mini blobs, or almost the same size, the second and third largest heat waves in our waters occurred in 2019 and 2020. So this seems to be a pattern that's getting definitely more warming in our waters coming up. So what does this mean for pyrosomes? Well, we did see, as I showed you, at least in a lot of our sampling, we didn't see a lot in 2019. They seem to have moved down south a little bit. We didn't get one of these cruises in 2020 because of COVID, but there were some surveys that went out in 2020, and they did see a few pyrosomes, but more to the south of Oregon. However, 2021's look, there's some pictures taken on the beach just in January showing large numbers of pyrosomes washing up on the beach right off of Newport here. So there must have been a lot out there. And there was even surveys that are going out now. There's been a couple of them that have seen them offshore, like 65 miles off the coast of Newport. They got large numbers of pyrosomes. So we may be in for another big summer of pyrosomes, although the good thing is it's going to be a La Nina year. So there was a tropical La Nina, which tends to be colder than El Nino years. So hopefully we may not get quite as bad as the last few years. So with that, I'd like to acknowledge a lot of the collaborators I've worked over the years with, including a number of people from University of Oregon that I'd like to give a shout out to for all their great collaboration, and Kelly's lab and Aaron Galloway as well over at OMB. So, and also I'd like to just acknowledge some of the funding and people that have helped out along the way. So with that, I would be happy to answer any questions, and I put my email here. If anybody doesn't have a chance to verbally ask a question and just want to email me, they're welcome to do that. But I want to leave you with this great picture of this leatherback turtle with consuming a huge tropical pyrosome. Fortunately, the tropics, or this species gets, fortunately our species here doesn't get it quite this large. I mean, ours gets maybe a little more than a foot long, perhaps, but this guy, there's one that occurs off of Australia that gets to be 30 feet long. And apparently turtles love these guys. So anyway, with that, I will be happy to open up for questions if I have time.
Thanks so much, Rick. We have 10 minutes for questions. Is that correct? It's 1:15. And then we also have like tea time. I always forget the tea time. When was that? Peter.
Sounds good.
We have 5 minutes for making tea afterwards, and then hang out, as long as people feel like, but usually until quarter till. So like half an hour.
Okay, great.
Okay, great.
So there are some questions in the chat. One is from Monica Rumimana, and then she says, "Thank you, Rick. Pyrosomes are fascinating creatures." Her question is pyrosome population growth also limited by nutrient availability for their food, like phytoplankton? Does nutrient availability also increase during the heat waves? And we all agree that the sea pickle name is pretty great for them.
Yeah. That's not something I'm particularly an expert on. Maybe Kelly could contribute a little bit more to that response. But anyway, I think that, they probably, having too many nutrients is probably not good for, in the water column is probably not good for pyrosomes, because that will lead to development of larger phytoplankton that I believe would clog their feeding apparatus, and they don't seem to do as well in very productive water. So that's why you tend to find these guys in kind of blue water oceanic ecosystems. I think that the nutrients were relatively low in coastal waters once the marine heat wave came through. So that was probably ideal conditions for them to develop. I don't know much about their reproduction or their growth. They add, what they do is apparently they add new zooids around the sort of, if you think of it as a cylinder, they're adding new zooids almost constantly, and that's how they get bigger and bigger. But I don't know if I know much about their reproduction. I don't know if I haven't been able to find a lot of papers on them, but I think they tend to bud off little zooids that become new colonies eventually. But maybe Kelly can, or somebody else can answer that a little more than I can, better.
I think those were great answers, Rick.
A little scattered, sorry. It's not my area of expertise, but
It's an interdisciplinary set of questions. Brings together a lot of different areas of oceanography and biology.
Yep. Yeah.
So should I move to the next question? There's another one in the chat.
Sure.
Yep. So there is Monica says thanks. And now Tyler Fountain has another question. Are the abundances of Pyrosoma atlanticum sampled in the Pacific Northwest between 2015 and 2017 comparable to the abundances in the more typical range of this species?
That's a good question. I think, well, I haven't seen a lot of studies that have actually gone out using the same type of gear to measure them or to collect them. I don't think there's been a lot of studies that looked at there have been some blooms of other pyrosome species, such as in the Indian Ocean and other places around the world that have been really high densities. But I think that ours may have been one of the highest ever recorded for this species anyway, partly because, we're even though the water tended to be a lot less productive than where they're normally found, the water tended to be a lot less productive than it's normally here. It was probably even a little bit more productive than the area that they're normally found. So there was probably a lot of food for these pyrosomes out there, and they were able to take advantage of that and just, grow very quickly. And but I think I don't know if I have a good answer for whether there's been other studies that have shown similar densities. They seem to be one of the higher recorded ones that have ever been made.
Yeah, Rick, I want to ask a question. You referred to movement several times during your talk, but I'm left curious as to whether or not that movement is the result of passive drifting with water masses that are associated with the extreme temperatures, or are they actually swimming to achieve more higher latitudes to get when they get up in Oregon? So swimming from south.
Well, I don't know whether they were able to swim, such long distance. They are capable of movements. In fact, there have been studies in the tropics that shows that they may migrate vertically up and down the water column up to 700 meters. So they're capable of, and we saw movement, and I think there was actually a student, Joanna Lyle of Kelly's, who looked at sort of vertically stratified samples and showed that they do move up and down in the water column quite a bit day and night. So they are capable of some movement, but I don't think that they're capable of swimming long distances like that. I think they were probably most likely transported in currents up here, or they just were residing in this water mass that got moved onto the coast and they came with it. So I don't think that they're able to move like, much like a fish or even a lot of invertebrates might be able to move. But they are capable of some locomotion, and some animals are able to change their location by migrating vertically into sort of different strata that are going in different directions. So if they preferentially want to move in one direction, they could move into an area that would be more conducive to that movement. But I think they're just generally part of this water mass that was moved from the tropics and offshore onto our shelf.
And in a related way, then, I presume their swimming is not sufficient to enable them to evade a predator when a predator attacks.
No, I don't think there's probably any predator they could probably evade. I don't think, no, I think that's what probably makes them very useful prey, because even slow-moving predators can probably capture them. But because of their size, at least during the summertime, a lot of things were not able to feed on pyrosomes. They were just too big to get inside the mouth cavity or something like that. We did see, we looked at a lot of other fish predators that occur off our waters, things like anchovies and sardines. And we showed that they actually were feeding on other gelatinous zooplankton during the marine heat wave, things like ctenophores and other small salps, but they probably didn't have the, you know, the mouth, they couldn't consume a large pyrosome like that. But I think that they're probably pyrosomes are probably not able to evade predators for the most part.
Just one more thing while we're on that topic. So from the fish's point of view, if they eat one of these, do they actually get any nutritional benefit? Are they capable of digesting it? Has anybody looked at that kind of question?
No, and that's something that I've talked to people about trying to do. You know, I mean, you would probably need to do that, those experiments in a laboratory, which can be done. I guess it hasn't been possible, but you know, you can look at sort of the evacuation rate of pyrosomes versus other things. They do get some nutrient off of them, I'm sure, but they're probably not ideal in terms of their you know, composition and things, and certainly not ideal compared to at least fish, smaller fish in terms of their caloric value. But there are a lot of fish that feed entirely their whole life on gelatinous zooplankton, and of course sea turtles do that. One of the largest fish out there, the ocean sunfish, gets to be, you know, huge, and they spend their whole life feeding on gelatinous zooplankton, and they can't swim very well compared to other fish. So that's perhaps one of the reasons why they were switched to things like medusae, that they can catch very easily and just eat lots of them and squeeze the water out, I guess, and get some nutrients out of it.
Thank you.
Mm-hmm. Thank you.
I've got a question if no one else does right now.
Sure.
So,
Joanna raised her hand.
oh, sorry.
Oh, you can go ahead. I can ask it afterwards.
No, go for it, Joanna.
Hey,
Okay.
Joanna.
Hi, Rick. It's nice to see you again.
Good to see you too.
I was wondering about, I think one of the fascinating things about this appearance of pyrosomes on the West Coast is, you know, between 2014 and 2018, there was a lot of pyrosomes off of Oregon. And then in 2019, they all just disappeared. And I was wondering if you could talk a little bit more about that disappearance in 2019 and if you have thoughts about what shifted over that short time period where they just disappeared.
Yeah, that's a great question, Joanne. I think, you know, we did see some indication that things returning somewhat to normal in terms of like things like copepod composition. We weren't, not me, but some of my colleagues were getting sort of the more typical northern copepod. So there probably was some change in the currents, and probably not. I think pyrosomes are probably still fairly prevalent offshore, but not. They probably didn't come onto the shelf or even, you know, anywhere close to the shelf as much in 2019, other than down in California. Now, they were, you know, that survey that I was telling you about, that they do that off of Central California, they had probably the worst pyrosome abundances they've ever had. I mean, they literally had to stop sampling on a lot of different locations. And, you know, I was emailing them and saying, wow, we aren't finding any of them here. We got one little tiny one here out of all of our, you know, compared to like hundreds or maybe millions that we caught in our sampling previous to that. So I don't know exactly what happened in 2019, but it was a little bit cooler that year, and things may have returned a bit to normal, at least in our waters, but not so in Southern California or Central California.
Thank you.
Yep.
So, hi there, Rick. Thanks for the great talk. I'm really curious about so do these things go away? Like, so the pyrosomes have disappeared from the coast of Oregon, 2018, 2019. Mm-hmm. Is it because they're offshore, or is it because they're just not present?
Well, 2018, they were certainly present, huge numbers. I could get back to that figure, but they were really abundant here in 2018. So 2019, I don't, you know, as I think I was just mentioning to Joanna, I think they were probably just further offshore. Of course, a lot of the ones from the previous year probably died and sank down the bottom or wherever, being consumed by things, but they just weren't in the coastal ecosystem. So there wasn't a lot of sampling going off around that I know of, sort of out in the Gulf of Alaska that summer. There was some during the winter, but I'm not familiar with a lot in the summertime to know if they were out there in large numbers, but they just weren't in the coastal waters.
And so has anyone tracked at a genetic level where these things are coming from, or described their population structures at all?
Not that I'm aware of. Maybe Kelly again might be able to tell you a little bit more about that. But I'm not aware of a lot of papers that have looked at the genetic population structure of these guys.
I don't think so. Yeah, not to my knowledge either.
It's super cool to do.
Mm-hmm.
Yeah.
Do you think it's just one species worldwide, Kelly, or are there subspecies?
I think it's possible that what's called Pyrosoma atlanticum could be more than one species. Hopefully we'll find out. We'll find out soon. We've been sending samples from different parts of the world to this aquatic symbiosis genome project. So it's focused on the symbioses, but hopefully we'll find more about the genetic structure of the hosts.
Cool.
Yeah.
I couldn't help myself, but the tropical ones, the Pyrosoma atlanticum that occur in the tropics, they're smooth. They have very different morphology in the colony, even though they're Pyrosoma atlanticum based on the soil descriptions. So.
Oh, are they? Yeah. There's probably different morphologies out there. Yeah. But it'd be interesting to see if they could figure out if this population originated in one particular region of the world, if it was, say off of maybe, Central America or more oceanic off of Hawaii or something like that. Who knows? But it's certainly interesting to see, there were a lot of other zooplankton that came from both southern and also sort of the Western Pacific region that came to, like copepods, for instance, that originated from off of Japan that they've only been seen prior. So there's a potential that these could have come from pretty far away, but it's really hard to say.
So seems that we can take 5 minutes for getting our teas or other drinks.
Okay.
and then we can come back here. Peter, we use the same link. Yeah.
Yep.
So we just turn on.
Okay.